Hello everyone, today we're going to be having a look at the DNA and a look at the structure and what makes up DNA. So DNA has a double helix structure, okay? So we can see from this diagram here, it kind of goes up this way, across, down here, and then it also has another strand that's running parallel to that. And they create this sort of helical structure, okay? Now if we zoom in a little bit and we have a look at each individual nucleotide, you can see here that a nucleotide consists of three things. This first light blue colored hexagon, or is it? It is a cytosine base. So that represents the base. Now this other thing next to it, this is actually the sugar. And the sugar and the phosphate. So the phosphate and sugar is this part here. That's the phosphate and sugar part there. That's what gives it the structure. So that DNA backbone is created by the phosphate and the sugar. All right. So DNA consists of these different colored bases here. Now these different colors, they represent different types of bases. So we have guanine, G bonds with cytosine, okay? And thymine, T bonds with adenine, A, okay? The way to remember that, cars go in garages and apples go in trees. Okay, A bonds with T, G bonds with C. Now there's also this bond in between them, in between the two bases, this area here, but that is a hydrogen bond. Okay, so hydrogen bonding occurs between complementary base pairs. All right. Now there's also a region of DNA called the primer region, or the promoter region. Now the promoter region is a series of bases called the tartar box. Now it's just a series of different types of bases. So TAT, AAA. This can signify the primer region, the promoter region of the DNA. There's other factors that is a promoter region as well, and that just signifies to the polymerase when it comes on to copy that DNA. It just signifies that this is where it needs to start copying from. It's that primer area. We'll go through that more when we talk about transcription and DNA replication. Now, there's also another thing that we will go through more, and that's called introns and exons. Introns are the bases that are removed from the RNA molecule when it goes into transcription. We'll talk about that more in future videos. So just for now, remember that introns are removed and exons we keep, right? And that's talking more about protein synthesis. So we'll go through that again when we talk about protein synthesis. But for the moment, just be familiar with the general structure of DNA.